if we want to know the composition, so what is there in blood? So what we have to do first? We have to take out the blood in a tube, say Wintrobe tube. So this Wintrobe tube will give you estimate what percentage of blood is constituted by plasma or cells. So what is the name you can remember? Wintrobe tube. W-I-N-T-R-O-B, Wintrobe tube, by which we can determine ESR, erythrocytic sedimentation rate, as well as PCB or hematocrit. PCB, hematocrit are same. Okay, hematocrit is 45, hematocrit is 48, hematocrit value is 42. What does it mean? If you take 100 unit of blood, out of this 42 unit is blood cells. Okay, RBC. Okay, so this is hematocrit. Hematocrit and PCB are same. Both are same information it is giving. So you have to know under which circumstance this hematocrit or PCB comes below normal or goes beyond. So these conditions you have to know. I already explained there to give you the examples. So WBC question also asked from this. Okay, name one condition, two condition in which PCB is okay, or in which condition hematocrit is high. What would be the consequence of increased hematocrit value, increased PCB value? What may be the consequence of just opposite, low hematocrit? So, study the books. I already told. Now, if we want to learn the composition, what we will be doing? We will be taking out blood into a Wintrobe tube. And the Wintrobe tube must have anticoagulants. Otherwise, blood will be clotted there. So, I will take blood and add in Wintrobe tube, which is having anticoagulants. You know, can you name the anticoagulants? Very good. One is EDTA. I told you to remember what is the full form of EDTA. So these are anticoagulants we use for this knowing the composition. Okay. So with anticoagulants, the blood is taken in the Wintrobe tube up to a mark. Okay. So you do one thing. In the lab, you will get one technician. Tell her to give you one Wintrobe tube.
Which lab?
This lab, hematology lab, next to this. Yeah. First. So I'll show you the hematocrit tube. Okay. So from the hematocrit tube, now it has to be centrifuged. It has to, here the tube will be rotated very fast for certain period of time. Okay, after that we will take out the tube, and here you will find how many layers? 3 layers. The top layer is plasma and bottom layer is cells. In between there is a white. What is there? What is the name of this? Name? Anybody? Write it down. Buffy coat. Buffy. B-U-F-F-Y. Buffy coat. And what you will get in this Buffy coat? Yeah. WBCs, platelets, and in abnormal conditions, abnormal cells like cancerous cells. Okay, the cells which should not be coming out of bone marrow into the circulation, that cells you may get. Pro-myeloblast. Example, primitive myeloblast. They are not supposed to come, but under this normal condition, means cancerous condition, they may enter here. And the virus, bacteria can enter within the WBCs, within RBC. Within RBC. Okay, some say amoebic. In case of amoebic pathology in the body, that organism can enter within the RBC. So RBC become parasitic infection. Now these parasitic cells, infected parasitic cells that you can identify in this layer. So this layer is very important, isn't it? So this Buffy coat is very important. Okay. So now I will very fast go into composition of plasma. After finishing that, then only I will come to the next part, that is different variety of cells, blood cells. Okay, so I'll move into composition of plasma. Okay, so this is the Wintrobe tube. Say, here zero marking starts from bottom. Okay, my focus is I have to know main part is what, cell or plasma? Cell. Means RBC. So cell will be there. So what is the reading? Say 4.5 means 45%. Up to 4 is there, means 40%. My PCB or hematocrit is 40%. What is the name of this tube? Wintrobe. Very good, excellent. And how can we, what I have to do before centrifugation? I have to add anticoagulant. It has to be, so wall should be, should have anticoagulants. Okay, then I will add blood. Or in a test tube, there should be anticoagulant, then I will add in the blood, mix it, then there is Pasteur pipette. What is the pipette? Pasteur, the narrow nozzle. Then I will suck. This is just like a dropper. So I will pull some blood, having anticoagulants, then I will insert it up to this mark, because the tube lumen is narrow. So I have to, with which tube you will be putting bloods in it? Pasteur pipette. Okay. Now I am entering into composition of plasma. What is there in plasma? Main component?
Water.
Huh?
Water.
Water. Very good. Main component is, see, if you are asked which component is abundant in plasma, what would be your answer? Water. And if I ask you what is the function of water in plasma, think it, I will ask. So water is there. What is this? It is water. How much water is there in plasma?
90 to 92%.
90 to 92%. On an average, 91%. Okay? 90 to 92% is water. Average 91%. And the rest 7% is solid. The rest 7% is solids. Okay? Now, this 7%, let's see what is there in this 7%. Okay, we'll find three things. One is solid component. Oh, sorry. One is organic component. Organic substance. Okay, what? Organic substance. Another portion is electrolyte. What? Electrolyte. And the third, last portion? Tell. Blood carries one of the important thing. Without this 3-4 seconds, if we are deprived of that, we may become faint. We may become unconscious. What is this? Oxygen. So, blood will contain oxygen, means gases. Maybe in physical solution, as physical solution, or as chemical compound. So, here what you will get oxygen, here you will get carbon dioxide, you may get nitrogen. Cations disease, problem with nitrogen. Deep sea divers. Okay, the gaseous, the dissolved nitrogen becomes gaseous form, and the blocks stores in joints in synovial fluid. So lot of pain, swelling. That is the symptoms of Cations disease. Okay, so gas is there. And here the electrolytes. Here two type of electrolytes you will get. Cations and anions. What are the cations? What are the cations? Positively charged ions. For example, sodium, potassium, calcium, zinc, copper. Okay, these are the cations. Okay, and there are also anions. What are these? Negatively charged ions. What are these? Chloride ion, hydrogen, HCO2, bicarbonate ion. Okay, chloride ion, sulfate ion, PR negative, meant protein ion. All these are, every ions, are very important. Slightest variation, more or less, you will be suffering disease. Slightest variation of sodium, electrolyte imbalance. Whenever there is electrolyte, the patients who are admitted in ICU, more often it is their electrolyte is imbalanced. If electrolyte is imbalanced, patient will be going to coma. There may be problem in cerebral function, there may be problem in cardiac functions. So all electrolytes are important. Okay? See, as I told, what happened? Tell me what happened. Pain. Why?
Neuroconnections.
Mechanical. I tried some mechanical pressure there and it created pain. Okay. Some receptors are there. Pain receptors are there. They were stimulated. Without sodium ion, this is not possible. These receptors were excited. When sodium ion entered within the cell, I disturbed the
balance.
No, I disturbed the cell, the outer cell membrane permeability. Normally, sodium ions are more outside the cell in extracellular fluid and less in intercellular fluid. So, when permeability changed, sodium channels are opened, then lot of sodium ion will be entering and the cell will be depolarized and they will produce action potential. And these action potentials are propagatory in nature and they will be propagating through nerves to his brain and he will sense in a pain area, somebody pinched me he is applying his eyes, he will have, sir has done this. Okay, great. So, sodium ion are important. Similarly, potassium ion are important. Chloride, all ions are very important. And you have to know, you have to learn what are their normal range. All ions. Okay, so when sodium ion more than normal amount, it is a pathological condition. Okay, less than normal amount, it is a pathological condition. Okay, there are some terms I will be coming to it. Okay, so here the electrolytes, both the electrolytes, either these are electrolytes, do it like this. Can you name one positively charged ion that is responsible for muscle contraction?
Calcium.
Right. Very good. Calcium ion. Without calcium ion, it's not possible. And for muscle contraction, you need energy ATP. And for this ATP, for muscle contraction, ATP has to be broken. And the enzyme is ATPase. And for this activity of this enzyme, one ion is required. One positively charged ion is required. That is magnesium ion. So without magnesium ion, is it possible? So all ions are very important for excitability, for muscle contraction, for maintaining what? The potential difference across the cell membrane. We call it resting membrane potential. Once we disturb the resting membrane potential by different type of stimuli, that will generate action potential, and this action potential will be propagatory, will be carried to the brain. And brain will now decide what to be done. So accordingly, action will be taken by the brain. So now I emphasized on this, every ions are very important. So you have to know. Sir told me zinc ion is important. What is the importance of, let's see, study your book. What is the importance of zinc? What is the importance of copper ion? What is the importance of, like this, all the electrolytes? Self-study. Everything cannot be taught in the class. That much time we don't have. So, you have to learn. If you want to develop expertise, you have to learn. No other alternative. You will develop confidence. Nobody can fail me. I have developed confidence in hematology. Whatever the question is, I will be able to answer. Otherwise, always there you will be in anxiety. What will happen? Okay, so develop. You have to learn by everything cannot be taught. So your mind should be inquisitive. Means what? Every aspect has to be learned. Why it is? What will happen if chloride ion is less than normal? What will happen? So this sort of question you ask yourself, and you will find very easy to get the answer these days. Google Baba is there. So ask your Baba, he will give you the answer. Isn't it? So that is the inquisitivity. Whenever you go through a line, a sentence in your book, when you find a new term, immediately you go for, what is this? And you will not forget this line again. So this way you have to learn. So, and now we are coming into, here you know the importance of oxygen, importance of carbon dioxide. It is the metabolic end product. It is not needed by the cell. It is the excretory product. It has to be eliminated from the body by means of respiratory process, expiration. Okay, it has to be carried to the lung, from where it will be. Otherwise, what will happen? Your cells are producing lot of carbon dioxide for generating ATP, utilizing, oxidizing the food glucose by oxygen, and you are developing lot of ADPs, and at the end product, you are getting carbon dioxide. What will happen to this? If it stays within the cell, then it will combine with water to form carbonic acid. It will be dissociated into bicarbonate hydrogen ion, and hydrogen ion concentration increase in hydrogen concentration within the cell. What will happen? Cell will become, what is acid? What is acid? What do you mean by acid? Really? You passed class 2 exam? Huh?
Something that releases extra ions.
Those who can give proton, give hydrogen ion in the solution, who can provide their acids. Okay. Hydrochloric acid, when it is dissociated, hydrogen ion, so this is an acid. Nitric acid, sulfuric acid, these are acids. And alkali, base, who can accept proton, who can accept hydrogen ion. Will you remember this? Always you remember. When is acid, means hydrogen concentration is more there. And it is buffered. Buffered means I added some acid here. So the solution must be acidic. But if buffer is there, when acid is added, I will find no, it is not. It has not become acidic. Somebody neutralize this acid. If I add or take out some hydrogen ion from here, it should become alkaline, but it is not alkaline. Somebody neutralize this condition by donating hydrogen ion. When I add hydrogen ion, it is neutralized by accepting hydrogen. These are called buffer. Got it? Got it, buffer? Got the idea of buffer? In railway, in between the rail compartments, what is there in between two compartments? Huh? One compartment, this is another compartment in between. These sort of things are there, isn't it? These are also called buffered. Buffer. Why these are called buffer? When brake is applied in that engine, what will happen to the bogies? They will, because of inertia, they will, some mechanical pressure will be applied on the other compartments. So, you are the passenger, you may be injured. You have experience. If suddenly your car brake is applied, what happens? You come forward and you may be injured. So that can be prevented by using the buffer. It will accept that mechanical pressure. That's why its name is given buffer. Okay, so in our blood also buffer is there. We have hemoglobin buffer. We have plasma protein, they also act as buffer. We have bicarbonate hydrogen ion buffer. We have phosphate buffer. We have ammonia. Different buffer system are there. They regulate, they acid base. They always maintain the pH at 7.4. If try within a range, you try to maintain this. Otherwise, if it goes beyond this, then there will be acidosis, then there may be alkalosis. These are the fatal condition if it is continuous. So these are the importance of pH. Got it? So, electrolytes, they maintain pH. They maintain the cellular activities by maintaining excitability. They are responsible for propagation of nerve impulse by saltatory or local loops. So here also depolation, waves. Through these waves, these are carried to the brain. And who are the responsible? These ions. So without that, this is not possible. For muscle contraction, ions are also, some ions are essential. Okay, so now conduction, weight, for every functions, ions are required. So these are their importance. Now we are coming into organic compounds. And these organic compounds are, one of the variety is protein. One of the variety is protein, we call it plasma protein, very important. What? Plasma proteins. And these plasma proteins are called colloid. They are called colloid. Why they are called colloid? Why they are called colloid? They are called colloid. Colloids are macromolecule. Macro. They cannot pass through semi-permeable membrane or parchment paper because of macrostructures. They are protein in nature. They are protein in nature. And these colloids are having charges on their surface. Protein, different types of proteins there, and their surface is negatively charged. Can they work together? Okay, we are friends, we will be going together. Can they? Why? You will repel me. I will repel you. Both are having negatively charged. I am not positively charged. As a result, all proteins are remain in suspension. They are not settled down. Okay, so colloids, proteins, plasma proteins are colloids, and they exert a pressure while in a solution. And this pressure is called osmotic pressure. What pressure? Osmotic pressure. Always remember, this whenever we will talk about osmotic pressure, it means it will try to retain some water with it. It has a philia for water. It wants water. Okay, so when there is osmotic pressure is high, it is more thirsty. It needs more water. When osmotic pressure less, it requires less oxygen, less water. So always remember this. Suppose your cell osmotic pressure is 25 millimeter of mercury and outside it is 30 millimeter of mercury. What will happen? Water. There will be movement of water either from the cell to outside or from outside to cell. Whose pressure? Whose attracting pressure for philia for water? Who is having more? Whose osmotic pressure is more? So within the cell pressure is less, so it will squeeze water from the cell. Cell will become shrink. Opposite can also happen. If outside osmotic pressure is less, inside osmotic pressure is more. From outside, water will enter within the cell, will infect, rupture. RBC may rupture. There will be hemolysis. It happens in pathological conditions. Okay, so proteins. These proteins, what is their normal value? They constitute 6 to 8 gram%. On an average, they are having 7 gram%. Okay? What is the normal value? If you take 100 ml plasma, and let's see how much out of this 100 ml, how much protein is there? Either range is 6 to 8 and average is 7 gram. And when you have less than 6, what is the condition will be given the term to this condition? Amount is less than 6. Anybody? If plasma protein concentration we determined in you, it is less than 6. What is the condition? Hypoproteinemia. Hypoproteinemia. These plasma proteins are synthesized in liver, except albumin, fibrinogen, these are produced in liver. So when I find there is hypoproteinemia, immediately I will have a problem in the liver. There must be some liver disease because these are synthesized in the liver. Or there is problem in the kidney. Kidney is filtering my plasma proteins along with urine going out of my body. Kidney disease. Either there is a kidney disease or there is liver disease. In this condition, we see hypoproteinemia. And when we find plasma protein level is above 8 gram %, what is this condition? Hyperproteinemia. When you can see this? Okay, AC. Hello. This bottle is having 100 ml of plasma. So this plasma is having plasma proteins. And what is the normal level? 6 to 8 gram %. So let's do one thing. I'll take out some water out of this. All other things are left inside. What will happen to the plasma protein concentration? It will increase or decrease? Yes, increase. It will be concentrated. No, only water I have taken out. Okay, now its concentration will be increased. Now what is the condition in its water is lost? Dehydration. So when you feel dehydration, your plasma protein concentration will increase. Isn't it? Think physiologically. Everything you will say, why it happened? What is the physiological reason? You have to explain. So everything is not discussed in the class. You have to inquisitive mind. You ask yourself, you try to find answers. It is there. Okay? So, there may be hyperproteinemia, there may be hyperproteinemia in different pathological conditions. I said you in my introduction, blood is, if body is functioning or having dysfunction, its reflection will be in the blood. If my kidney is not working, if my liver is not, it will be reflected in my blood. And if blood, in other situation, if blood concentration of some components is changed, body function will also be changed in both ways. So that's why it is very important. By studying blood, you can say whether it is liver disease or kidney disease or any other disease. So you have to be analytical power, you must develop. This, because it will give you the window. You are here and this is the window. What is happening there? You can see through this window. This window is blood. And they are the other organs outside. What is happening on the other side, through this window, through these blood levels, composition, blood composition, you can assess what is happening in outside. So that's why it is very important to you. Okay, and now these plasma proteins are of 3 types. In some books you will get 4 types. Okay, what are these? What are these? Different variety of plasma proteins. Bole kono plasma protein nahi. Tell the name. Anybody? I am not asking you. Wow, very good. I will ask you later. Anybody? Albumin. Wow, very nice. One is albumin. Another is globulin. Another is. These are required for blood clotting. Without this, clotting is not possible. Prothrombin is what? Fibrinogen. Okay? Fibrinogen is required for blood clotting. And fibrinogen content in the blood is increased during some pathological condition, injury, burn, fibrinogen content will rise in the plasma. A case, you go through a burn case, check his blood plasma protein level, you will find fibrinogen level is increased. It has got some problem. It has got some problem. Higher the fibrinogen content, the RBCs cannot remain in suspension. They will be settling down. High fibrinogen content, low suspension stability. Low suspension stability. So this will increase ESR. So in such case, you will find increased ESR. Cannot? Only because there is rise in fibrinogen level. Okay. So albumin. So plasma protein, 3 or 4 types. One is albumin, another is globulin, another is fibrinogen, and other is prothrombin. And these two are involved with coagulation of blood. Coagulation, is it good or bad? Is it good or bad? Cut, blood coming out. Is it good? Not good. It is good. It is protective in nature, protective function. It protects you from hemorrhage. Got it? It is protective. It is not bad. What will happen if blood doesn't coagulate? Because of hemorrhage, the patient may die. So because of this protection, there is no heavy blood loss. Okay. So let's say albumin. What? Definitely it has got some functions. What are its function? Let's see. One is maintaining. Okay. Osmotic pressure. Osmotic balance. What I'll do with this? What I'll do with this? Osmotic balance. So what is the use of this? What is the importance? What is the significance of this? Everything you must know. Why blood is maintaining osmotic pressure? See, I am giving you one example. Now you will realize its importance. Your cells, body cells, they are getting nutrients, all the required things necessary for the functioning of the cell. From where it is getting? Artery or arteriole or capillary or veins. What? Capillary. So, this is the cell and this is the capillary and this is the also capillary. This is the venous end. Venous end. From here it will combine with other venules and form a vein. The arteries, arterioles, they will be branched into with various capillaries and each capillary will be supplying the cells. So this is the arterial end of capillary. So blood flow will be from this side to that side or from this side to that side. This very good from this side to this side. And this, what is flowing through this? Blood. Definitely it will be having some pressure. Huh? And this pressure is called hydrostatic pressure or blood pressure. What is this pressure? How much pressure is there? 37 millimeter of mercury. Right, 37 millimeter of mercury is the arterial pressure present in arterial end. Okay? And this blood is having plasma protein. They are exerting a pressure. What is, how much it is? 25 millimeter of mercury, right? 25 millimeter of mercury. And these two pressure, no, they act one opposite to each other. Their action is opposite to each other. A pipe, water pipe. If you find the leakage, what will happen? What will coming out with the pressure? That is the hydrostatic pressure. Okay, so hydrostatic pressure is the driving pressure. It wants to eliminate fluid out of the vessel. And our osmotic pressure, they prevent it. They prevent it. No, don't go. You be with me. So, osmotic pressure tries to retain the water, whereas hydrostatic pressure wants to eliminate two outsides. Okay, fine. Here it is 37 mm of Hg. And colloidal osmotic pressure here, how much? 25 millimeter of Hg. So it is the pressure wants to collect and that pressure wants to push out. Okay, now there will be balance between the two. Who is more powerful? Hydrostatic pressure. As a result, what will happen? And these capillaries, no, this weight, these capillaries are having endothelial cells. Single layer of endothelials. One endothelial cells, another. So this is one layer and this is another layer outside. This is the lumen. Now these cells are not tightly attached to one another. There is a gap. And this gap is called fenestra. Endothelial cell gap. Okay? When your body becomes affected, means inflamed, this gap increases. Okay. What is the use of this? Because of hydrostatic pressure, 37. So I am to push my water through this to the outside, and these are the sieve. Everything cannot come out. My cells cannot come out because this is narrow. Cells are bigger, so they cannot pass. Plasma proteins, they cannot pass. Their size is more than this diameter. Only some ions, vitamins, minerals, hormones, they can pass. Water, they can pass. Okay, as a result, through this, the required substance, glucose, amino acids, whatever the cell requires, everything will enter from blood vessel into here. Okay, because of this difference, what is the difference? 37 minus 25. How much? 12. Because of 12 millimeter positive pressure, fluid will be coming out. Okay? Now, the cell got whatever it is required, everything for, okay. I got glucose, I got amino acids, I got hormones, I got everything. So everything will be entered here, diffused in. Now the cell is having some excretory product, isn't it? So what will the cell will diffuse it out into the ECF. Okay, so exchange will be there. The required thing will be taken by the cells and the unnecessary things will be given out. Now what will happen? When this blood enters here, this pressure falls, it is widened. As a result, pressure will fall. It is 17 mm of Hg. Whereas protein is not coming out, it is 25 millimeter Hg. Still, now what will happen? Drawing pressure is more. With that one is 17 only. What is the difference? What is the difference? How much? 8 is the difference. Okay, so 8. So because of this 8 negative pressure, 8 millimeter Hg, the fluid will enter here. More fluid entered, less came out, less fluid because pressure is this. That one was 12 or 11 and it is 8, 7 or 8. Now more fluid has entered into the ECF but less fluid has came back into the circulation. So in the tissue space, some fluid will be retained. Under normal condition, what will happen to this? If it continues, then the ECF will rise and swollen. Okay, but normally what happens? This extra fluid is taken back by lymphatic channels. Through these lymphatic channels, again this will be coming back to the circulation. Now you see one picture. Plasma protein concentration fell down. Osmotic pressure will be reduced. Osmotic pressure will be reduced. See the case. It is now 20. What is the difference? 17. Now lot of more fluid will be coming because difference is more. And here what will happen? It is 20. Only 3. Very less amount will be coming. And gradually lot of fluid will, all the fluid will not be taken by the lymphatic channels. Lot of fluid will be accumulated there and this is called edema. And this is called edema. There will be edema, swollen. If you press, it will be like this. I'll show you one image. Now you got the importance of maintenance of osmotic pressure. So when plasma protein synthesis is affected by the liver, somebody taking a lot of alcohol and damaged his liver, what will it do? Will it produce sufficient plasma proteins? No, there will be edema. There will be accumulation of fluid in his peritoneal cavity, in his extracellular space. Understood the importance of it? So every aspect has importance. Okay? Now, the globulins. Some albumins has other function. Other than this, they are carrier. Some hormones, some vitamins are carried by the albumin from one place to other. You have taken food, these are digested and it has to be taken to the liver or to the cells. How they will be taken? The albumin will be helping. Okay, come with me. I'll take you there as carrier. Okay, so they can act as carrier. Globulin, these are also carrier. In my Saturday class, I will discuss in detail. Globulin, immunity. It provides immunity to your body. Different types of globulins are there. Alpha globulin, beta globulin, gamma globulin. Gamma globulins are antibodies. Alpha and beta, they are carrier protein. They are also act as carrier. We will be discussing it later during our coming class in detail. And prothrombin. Prothrombin is a proenzyme present made of protein. Proenzyme, when it activates, it is activated by by coagulation factors. It produces thrombin. Now it is an enzyme, and it will now convert fibrinogen into fibrin. And fibrins are thread. Initially they are insoluble, and by one plasma coagulation factor they become insoluble. Now your platelet, your platelet, your platelet, their platelet, they all, the platelets will be, they will be tied by the fibrin thread. So you will make a clump, you will make a clot. Okay, so clot is formed mainly because of platelets and involvement of, initially what happens, we call it, what is the difference between homeostasis and hemostasis? Hemostasis, by which blood loss is prevented. By which blood loss or fluid. It is the initial step of coagulation. Hello? Blood vessel. I injured it. Okay? Blood will start to come out. Now what will happen? First step. The vessel will be constricted locally. There will be vasoconstriction. The injured blood vessel will liberate something. The platelets will liberate something, and they will initiate vasoconstriction. So, when blood vessel is constricted, so there will be less flow. So, blood loss will be minimized to some extent. Now, rest part comes. The platelets will adhere to the injured tissues. When they are adhered, they become activated. When they become activated, they become aggregated. You will bind him, you will bind him, you will bind him, so you will become aggregated. When they are activated, they release something. They release something. Because of this release, ultimately fibrinogen will be converted and they will be bound together and tied by this thread. So, platelet plug will be formed at the injured area. Temporary platelet plug. As a result, it is not clot. It is not, it is the platelet plug, initial part. So, because of this plug, blood cannot come out. So, to some extent now prevented. But it is not permanent. Now it has to be made permanent. Then coagulation starts by intrinsic and extrinsic pathway. As a result, some RBCs will also be entangled there. And a solid, tight plug will be formed. Permanent plug will be formed. So initial vasoconstriction. Remember everybody. Initially when injured, what will happen? Injury, vasoconstriction. Then platelet adherence to the injured area. Now because of adherence, the platelets will be aggregated. Now they will secrete. All these platelets are having some granules inside. All these granules will be secreted and they contain lot of important things. With this, they activate coagulation process. Intrinsic path, extrinsic path is started. Now, fibrin is formed, they are tied, permanent plug is formed. So, after that permanent plug is formed, no more blood ejaculation stopped. Ejection stopped. Now, what will happen? They will be staying there permanently? No. They will be fragmented. And what will happen to this fragmented? That will not enter into circulation. They will be taken up by another variety of WBC called macrophage. They are nothing but neutrophil. Remember this thing. Neutrophil, they can come out of the blood vessel by a process called diapedesis. So with this diapedesis, they will be coming out of the blood vessel And what they will do? Convert into macrophage. Now, this macrophage Rakhosh with fragmented blood clots are engulfed, digested, removed. And, okay, clot is removed. Now what will happen to the injured place? Injured place will start to develop, to heal up. The platelets and the wall of this cell, they will secrete some growth factors. So because of these growth factors, the cells will start multiplying. Gradually this will be healed up. Okay, very interesting, isn't it? So we'll be starting more detail. Okay, so albumin, fibrinogen, prothrombin, these are. Now other organic substances. I will finish again. I have taken 1 hour. Still it is not completed. Or with cells, I couldn't enter in cells. And actually when I talk, I try to explain every aspect so that you can take this, you can remember this, you can know that behind what is the reason. So that's why I take a lot of time. But how much I give, that you should remember. Okay. So what are the other organics? We take carbohydrates, rice, bread. Why we take it? These are complex. These are needed. We take it for glucose, but glucose is simple form. This carbohydrate starch, these are complex form. Our blood, they cannot enter in blood. So it has to be made simplified. Similarly, proteins are complicated, complex structure. The cell need amino acid. They don't need protein. They need amino acid. And we know amino acids are present in protein. So that's why we take fish, meats, eggs, milk, we take. Now these are complex, no? So it has to be made simple into converting to amino acids. Fats, we take fats. So these fats are again complex. So it has to be made simplified into fatty acid and glycerol. How they will be doing this? How it will become simplified? That's why we have got a system, digestive system, function to simplify all these complex material into simpler form so that they are now absorbable. So glucose is needed by the cell. First they will be entering into the cell, into the blood. Then they will go to the cell. So it must be a component of organic component glucose. So amino acid must be a component, fatty acid must be a component, fat must be a component, lipoprotein must be a component, vitamin must be a component, hormone must be a component. And your cells are producing lot of non-protein nitrogen, lot of excretory products. And what are these products? Urea, uric acid, creatine, creatinine, xanthine, hypoxanthine. These are all excrete. We don't want this. So blood, from the cell it is entering into the blood. So there should be their presence. You can estimate this, whether blood cell is producing or it is not, kidney is not working. So what will happen in the blood? Their level will be rising. So by increasing, if you find somebody's blood in plasma, there is increased level of creatinine, increased level of urea. What do you think? Kidney is not functioning properly. Okay. So these things are present in blood. Okay. So organic compound, this organic compound, and there are vitamins, minerals, as I said. So these are the various components of plasma. Have you got it? Okay.